Up to you, I'm not going to. Hello, everyone. So it's really great to be with you tonight. Thank you for coming and listening to me talk and giving up your evening. My name is Ben. I am a PhD student, and I'm a PhD student with a bit of a double life. When my friends ask me what I do, what is your PhD, they ask, and I say I am a chemical biologist, and they say, well, what on earth does that mean? And basically, I have, as I say, this double life. I work at the LMS. I have a group there doing biology, and I also work in the Department of Chemistry at Imperial College, where I also am a chemist. I'm a chemist by background, but we basically try and use our chemistry to answer biological questions, and it's great fun getting to work across lines and getting to do all kinds of different things. So, as you see, my talk is the shape of you. So the first question I know you want to answer, no, I'm not going to sing the song. I'm really sorry, it's just not going to happen. You don't want to hear it. But we are going to be looking at the shape of DNA over the sound of traffic. So we're going to be answering a series of questions this evening. We're going to be asking, what does DNA look like? We're going to be asking, how do we look inside cells and look at DNA? And then, what does the shape of our DNA mean for us? And I'm going to need all of your help to answer some of these questions. Are you willing to give me that help? Okay, so I think we all agree that was a poor response from everyone here, but it's Monday, I get it, it's been a long day, you've all worked hard, so I did think that this might happen in advance. DNA matters to everything that DNA is going to do. And all of our cells machines need to look. DNA has the code programming everything we do. And our cells machines need to go in there and need to be able to read it. And the structure and shape is super important to being able to do that. This comes from an animation of DNA being replicated. And those machines that are stuck to the DNA chains, they need to be able to hold on to and recognize that shape, that double helix. and it's crucially important to how it all works. So the shape is really key to how. If anyone's warm, you can complain later. So the shape is really important. So the double helix. So we have two strands. How do they stick together? Does anyone know? Let's touch on the one.
Hydrogen bond?
Yeah, so bonding between different bases. So, that's why you got it. So, you might have heard that DNA has this code, and we represent this code with 4 letters. And those 4 letters are A, C, G, and T. But really, obviously, they're not letters in DNA, they're different chemical structures that we just talk about as being those letters. And they have their favourite friends. So A and T are friends, and they like to stick together. G and C are friends, and they like to stick together. And this is an important part of how DNA takes on that double helix shape. But we're going to talk about a different shape, because not all DNA is double helix. There are other shapes DNA can take. Most shapes that DNA can take that aren't double helix only are really happy existing at really wacky, extreme conditions, heated up to something, cool it down to something, put it under high pressure. But there is a structure that's stable at the same conditions are in your cells. And we're going to learn a bit about that. And it's called a quadruplex. And this is the thing that I'm interested in studying. And to talk about it, to introduce it, we're going to have to talk about one of those bases, one of those letters in the code that I told you about before. And it's called guanine. And I am going to need a volunteer. I'm going to wait until there is a volunteer, so this is up to you. Kaylin, come up here a bit. Well done, Kaylin. You can have a budget at the end. Okay, so while Kaylin's coming forward, Kaylin is going to be a guanine. Now, Kaylin as a guanine, what she really wants to do is bind as a G to her best friend, cytosine. That's her favourite other DNA base to bind to. But Kaylin can do that, and she can bind kind of head to head like this. So Kaylin, you put your 2 arms out like this. So if I were a cytosine, we could come and we could happily, this is how we like to be, this is how we like to pair up. But guanine can also do something else, and this was discovered way back when, in the 1800s. That if you just have guanines on their own, they can do something else. Now, I'm now going to need more volunteers, so some of you are going to have to volunteer. And that something else is critical to forming this, which is the quadruplex. So we have our 1 guanine. Kaylin, you are our 1 guanine. Can I have another guanine? Come on, thank you very much. Everyone who comes up gets a punch. Okay, so with a cytosine, you can do your nice face-to-face bonding, but you're not a cytosine, you're a guanine, okay? So what they can do is they can bind, but basically to the side of the other guanine. So if you stand like that, okay, if you turn this way, and you are going to bind to the shoulder of the next guanine. So instead of binding kind of face-to-face, the guanines are bound shoulder to shoulder, and chemically we can represent that like this. So you can see that guanine is kind of on its side. But that means that you have your arms free, right? You could bind to something else. So what if we had another guanine here? Another guanine, anyone? Yeah, come on. Right, so you can bind to this guanine's shoulder. Well, you're going to keep your arms in front of you. Okay. If you could face that way, okay, and then you can bind to this shoulder. So now we have 3 guanines all bound together. We can see the next guanine here. You can see the guanine there, kind of upside down. But obviously, you as a guanine, you also have arms to bind to something else. Okay. So you could bind to another guanine, right? So who else wants to be a guanine? I only need 1 more of you. Okay. Tiffany, you can say. So you can bind onto the next one's shoulder. You're getting almost ahead here. Okay, so now we have 4. But obviously, our final guanine also can bind, and we're now sort of in a little square here, aren't we? You can bind back onto Kaylin. And so this structure of 4 guanines, you can now see that. So this is what it looks like chemically, can all bind together into what we call a G-tetrad, this little square of guanines here. And so this was actually found all the way back in the 1800s. Guanines can self-assemble into nice structures. Now, that's one part of the quadruplex. So you can see this is a little cartoon quadruplex on the left here. And this shape that they're representing is one of those flat squares. And what happens is that these can stack on top of each other. So if health and safety were no issue, I would now get 4 more of you to sit on their shoulders and do this again. But unfortunately, I've got to look after you. So what we can get is 2 layers of this, 3 layers of this, maybe even 4 layers of this, all stacking one on top of the other, forms us our G quadruplex, is what the structure is called. Thank you very much, Guardians. You can go sit down. Round of applause for Guardians. So, rather than having these intertwined double helical structures, we can have these quadruplexes. So to give you, this is what, if we fire X-rays at a quadruplex and kind of look at the structure that way, we get a shape that looks kind of like this, which is a bit kind of mesmerizing, it's a bit kind of, it's almost a bit hypnotic with the spiral there. But it's easiest to think about it in its cartoon form over here. And so you can have, as long as you have all these guanine bases, these Gs, they can build this quadruplex structure. And they can look like that. And the way I've drawn it there, you can see it's one piece of DNA wrapping around itself. You can actually have it so that it's 4 separate pieces of DNA. But more likely inside a cell, you're going to have the one wrapping over itself. And it can kind of look like this. So you have your double helix, that's kind of the normal DNA. But then if you have a sequence which has lots of guanines in it, it is potentially stable for it to fold up on itself like this. And the way we often talk about this is if you imagine that the double helix is like, you've got your 2 pieces of DNA and they're kind of wrapped around each other. A quadruplex would be like having a knot on one of those. That is our DNA knot that I talked about in the bio for anyone who followed me. And so you might imagine that if you have an odd knot, we talked about all those cellular machines earlier that are in your cell doing stuff. Well, having a knot is going to really affect how they're able to interact with the DNA. And you'd be absolutely right. It's predicted that quadruplexes inside a cell would affect all kinds of processes. Someone's drawn them up here in a nice paper. And we'll get to some of these. But the key question you're probably wondering is, Ben, we've gone on about this for a while now. And it's all very interesting. You've got people standing around the square. But does this actually matter? Does this happen inside a cell? Does this happen inside me? Well, looking at our cartoon cell here, trying to find, we're now going to look at how you can look inside a cell. Because trying to find quadruplexes in a cell is going to be quite tricky. Because there's a lot of DNA, and if there are quadruplexes, there aren't going to be many of them. It's kind of like trying to find a needle in a haystack. And so we have to work quite hard to try and go and look for them. So how do we normally look inside a cell if we want to look inside a cell? Well, we normally look inside a cell using something called fluorescence, which is basically we get it to light up. I'm now going to quickly use all of you, you're all volunteers, sorry, to demonstrate fluorescence, okay? So you are all fluorescent molecules now. Well done, you've successfully transformed into fluorescent molecules. When fluorescent molecules have light shone on them, I have a light on the end of this pen, I'm not going to shine it at you yet, because I don't want the response to happen yet. When the light shines on you, you get excited. You absorb the energy and you get all excited. So when I shine this light on you, because you're fluorescent molecules, you're all going to get super excited. I want to see that, okay? Okay? Right, and then you all, I didn't even have to cue you for this bit. So you all got super excited, but you didn't stay excited for that long. You relaxed back to your normal sedentary sitting down state. And in your case, you got rid of that energy of excitement by probably becoming slightly warmer, sweating a little bit, doing something like that. In the case of fluorescent molecules, they release a little packet of light called a photon. And so that's what happens with these. So for these kinds of images of the cell, we have a molecule that will go and bind to something in the cell that we want to look at, and then we'll shine light on it using often a laser or a lamp or something, and it will get excited, and it will then relax back, releasing a photon of light back out at us so we can see where it is. And then we can find different stuff in the cell. But that involves us having a molecule that can bind to what we want to look at specifically. And we're trying to find a needle in a haystack. And not only are we trying to find a needle in a haystack, we're trying to find a needle made out of hay in a haystack, because our quadruplex is also DNA swamped with other DNA. So we need something a bit cleverer, a bit smarter. So normally we use something called fluorescence intensity. So we're just looking for our fluorescent molecule here to release a little packet of light, a photon, and then we see it. That's what we're normally looking at. But we can do something a bit cleverer. All of you, when you got excited just before, you didn't all stop being excited at exactly the same time. You did it at different times. That's something we call fluorescence lifetime. It's how long you stay excited for before you relax back down to your normal state. And that's what I use. And fluorescence lifetime, instead of just looking for when the little packets of light come out, we look a whole bunch of times at different time intervals really, really quickly and see when those photons arrive at us, to see when those molecules are releasing their little packets of light. And when they release their packet of light depends on what's around it. So we can use that to tell what the molecule is stuck to in our cell. And what we end up seeing is something like this. We see a number of little packets of photon light over an amount of time, and the average amount of time it takes us to see the light is our fluorescence lifetime. And this is what I use. This is what I use in the lab. So how can I use that to find our needle in our haystack, to find our quadruplex? Well, we use something that I showed this written down to my girlfriend recently, and she was just like, I don't even know how you would start saying that word. We use something called trianguleniums, which is a very fancy word of saying molecules that look like triangles. Specifically this one, which has been given the name Dotron 2. It's a triangular molecule. That's all you need to know about that molecule for now. And it's a fluorescent molecule, so it gets excited. But it has an interesting property to do with its lifetime, which is it can go inside cells. So we'll have our cells, we'll have our triangulenium molecule, which for now we're just going to represent by this triangle, and it will go inside the cell. And it will bind to all the DNA, and when we pulse a laser at it, we shine light at it, all of these molecules will fluoresce. They'll send photons of light back out at us. But they'll send out photons of light at different times depending on what they're stuck to. The fluorescence lifetime will change. And it turns out that if the triangulenium is stuck to the quadruplexes, then it will fluoresce for a longer lifetime than if it's stuck to any other structure of DNA or anything else in the cell for that matter. So that gives us some new tool we can use. We're not just looking for the photons, we're looking for when they arrive at us. And we know that the longer they wait from when they get excited, they're more likely to be coming from these quadruplexes. And so when we've tested this, we know that after 8 nanoseconds, we can see, on average, the photons coming from double helix DNA. But after 14 nanoseconds, which to all of us is not much longer, but to these photons and these molecules is plenty of time, we're on average seeing coming from these quadruplexes. And so this gives us a new tool we can use to try and find quadruplexes inside cells. So what does this mean practically? When I'm in the lab, what does this mean? Well, it largely means sitting in this, the dark. Actually, this is just a black box on PowerPoint. This is not this dark, it's this dark. This is my lab. This is our whole setup and our microscope. So you might be able to spot, I don't have a pointer, but there. You might be able to see there's a microscope. And then under this big box, we have our lasers. Because what we need, I just said before, we are looking at lifetimes of nanoseconds. So we need a laser that pulses really, really quickly and is really, really intense to excite all of those molecules very quickly. So we have a laser that will pulse, and it will pulse 20 million times a second, which is a lot, plenty, some may say. And we will use the microscope and collect all the photons, but not only will we collect them, we'll know when they arrived at us compared to when the laser pulsed. And so what does that mean we actually get in terms of, you can see my laptop over there, what actually pops up on my laptop after I've used all of this? Well, we get an image like this, which is not the prettiest. For anyone here who does microscopy, this isn't the prettiest image, because we can't use a high resolution for various reasons that I'm not going to go into now. But we collect an image like this. You can see lots of little cells and lots of the nucleus of the cell where all the DNA is. And in every single pixel, there's 256 times 256 pixels in this image. every pixel, we get the time that we saw that light arriving at us. And as we said before, we can correlate that to what the molecule was stuck to at the time. And so we can look at the ratio of the amount of double helix and quadruplex that we're seeing. And we can try and work out how much, if there is quadruplex there, and how much. So some of you who are more scientifically minded might now be going, well, Ben, how do you know this works? Show me some evidence that this works. I will take that challenge. So if we have these molecules and we just put them in with some DNA, forget all the cells for the start, let's just put it in with some DNA in the equivalent of a test tube, a cuvette, then we know that we see these different lifetimes when there's no cell there at all. When we do have a cell, we know that if we have our triangulonium stuck to our quadruplex, and then we throw in a molecule that binds quadruplexes really, really, really strongly and in exactly the same way, that it throws away the triangulonium gets pushed aside, and we see the lifetime change. It's called a displacement. So that's pretty cool. And we also know that if we have our cell and we do something to perturb it and shake it up so that we predict it would then be more likely that quadruplexes would form, well then we also see our lifetime change. And this is something I've been doing quite recently, is messing around with the cell to do something to try and make more quadruplexes happen. And I can actually see the fluorescence lifetime of my molecule changing when I do that, which suggests that there not only are quadruplexes in there, but I can make more happen and see the response. There's our triangulonium going in. So the final question that I said we would answer today is, what does this mean for us? And for that, I have to go, this is a journey I actually do on a very regular basis. I am represented by this icon of a person. I am at the chemistry department, the new chemistry building in White City, and I will go running off from my lab with that laser that I showed you before, off to the LMS at Hammersmith Hospital, where my kind of biology group is. And what we do in that group is we look at a specific part of the chromosome called a telomere. So this is a cartoon chromosome. And telomeres are caps that protect the end of the chromosomes that are in all the way the DNA is packaged in the cells. Telomeres are these little caps, and you can think of them like the caps that are on the ends of your shoelaces. So those caps are there to stop the ends of your shoelace from fraying. These caps are there to protect the ends of your chromosomes, because as time goes on and your cells copy their DNA, they tend to lose a bit at the end each time. And so if you didn't have caps protecting, you would just lose all your very useful DNA, and that would be problematic for your cells. So the little caps there to protect. And they're caps, they're made of DNA, and they're made of a very repetitive bit of DNA. It just so happens to have a lot of Gs, a lot of those guanine bases. We're back to our guanines here. What can those guanines do? They could self-assemble with each other, like we had our people doing here, and they could form quadruplexes. So we could have little quadruplexes at the ends of our chromosomes in the telomeres. And that would be really interesting. And I'm going to give you only one reason why that might be interesting. And it's the reason why a lot of people got very interested in this when kind of the quadruplex field was first kind of getting up and running. And that reason is the same reason that motivates a lot of research, which is curing cancer. Because what are cancer cells? Cancer cells are cells that will rapidly divide and take over. And in order to do that, they have to be immortal. But these telomeres actually kind of set a limit on the mortality of your cells. Because over time they'll degrade, and when you have none left, it kind of puts a clock on how long your cells can live. But cancer cells get over that, and they get over it using a special protein called telomerase. And telomerase, represented by this blue circle, will basically go to the telomere, and it will go along the telomere and extend it and make it really long so that the cell can become immortal and live forever and take over everything, which is good for the cancer cell and bad for us. So what people first thought in the quadruplex field was, well, if there are quadruplexes in these telomeres that they can form, then they could get in the way of this telomerase enzyme. It can get stuck. It can't do its job. We can make these cancer cells mortal again and kill them and cure cancer. About 90% of all human cancers use this kind of telomerase method for helping to make themselves immortal. Now, people have tried to do this by making things that are going to force quadriplexes to form these telomeres. It hasn't quite proved as simple as they had hoped originally. But there are lots of people still trying to do this. And if we can have a method to study quadriplexes and see what they might be doing inside living cells, which is what I've been trying to do, then maybe we can have a more sophisticated understanding of how to go about doing things like this. Or even, I showed you this picture earlier, there's lots of other ways quadriplexes could be affecting your cells. So to use the code to make it into something, you have to copy it from the DNA inside your cell nucleus so that it can go off and be used somewhere else. Well, if there's quadriplexes there, maybe they could get in the way of that copying. Or maybe even they could provide a signal, a signpost almost, to indicate that that's something that should be copied. There's also it's been shown actually that these sequences which we think are likely to form quadriplexes tend to be overabundant in places where we would start copying a specific gene to be expressed. So that's an interesting possibility that the cell is using this as a tool. There's lots of other ways. It could also be helping with controlling how our cells replicate their DNA and divide. Quadriplexes could be getting in the way of that process as well. It was originally commented a while back that if the quadruplex can exist inside a cell, then nature surely has found a way of using it to its advantage. And now we are trying to find a way of studying it so that we can maybe use it to our advantage and use it to hopefully, at the very least, increase our understanding of how our cells work, if not find new ways of intervening and doing cool things, curing diseases, etc. So we're just taking that first step along that path. Science is a team game. I'm here presenting, but lots of people have helped me, and especially being someone who crosses disciplines, I need all the help I can get to be able to do everything. These are the people who directly helped me with bits of work, but there's loads of people in all three of my groups in both of my departments who've done those to help me out. So thank you to all of them. And thank you all for coming, and thank you all for listening. And I'd happily take any questions. And thank you for volunteering.